topic is the BTK inhibitors and mantle cell lymphoma, but I wanted to show some slides to put the whole thing in a context. So you notice my slide has some have the moon. So this represent the MD Anderson moonshot program. So what do we do in the moonshot program? The goal of the B cell moonshot, which I'm heading, is to double the cure rate of B cell lymphoma from 30 to 60%. You probably, the people from the US know that close to 1 million Americans live with lymphoma, and 90% of those lymphomas are B cell lymphoma. So for such a common malignancy, the current cure rate is only about 30%. So, so our goal is to double the cure rate from 30 to 60% for in 5 years. A lot of people think, Dr. Wang, you're crazy. For one institution, this is overly ambitious. This is impossible. But let me show you some of the published data from MD Anderson. For both southern patients, for this published, so for both indolent and aggressive lymphomas, in the past 6 years, almost every decade, there's a steady progression of a median free survival. And so our aspiration to double the cure rate is based on data and knowledge. And this was achieved from little technology novel therapies that we have today. Our generation is equipped with much more technology than novel therapies, more than anybody in the history. So we should be able to achieve this in faster pace. So in order to do this, we have a very vast program. We have 3 flagship programs, and we have groups on each flagship: novel targeted therapies, cellular therapies, immunotherapies. We have dedicated tissue bank, hematopathology. We have platforms to sequence genes, RNA, DNA at MD Anderson, proteomics. So we work in harmony. We also have a basic investment in the basic and translational sciences. So today I'm going to focus on the BTK inhibitors. You saw these slides again before. Ibrutinib is by far the most developed BTK inhibitors. Single agent activity 67%, and this is the data that served the basis of clinical approval by the FDA. And this is to show you the power of the small molecule. Just one pill, 1 day for only 60 days. The median nano mantle cell lymphoma mass was shrunk to almost over 90%. So there's a real power in small molecules. And this is another clinical trial called the SPARK study, where we treated also 110 patients, and we also have similar responses of like 64, 67, 3%. And then we have responders and not so good responders. We find many mutations in the non-responders and the intermediate responders. And we find that these mutations are in 4 class of mutations, namely the BCR, the epigenetic modifiers, the PIM, mTOR, and the oncogenes. So this mutations also is associated with different survivals, because if you have different mutations, their survival is poor in terms of the NF-kappa B mutation pathway. And also those mutations along the epigenetic modifiers also predict our survival. We are actually in the middle of doing functional genomics to for if you put in this gene vector with those genes and certain genes, the cell lines lose the apoptotic apoptosis, become resistant to the ibrutinib. So we hope that we can share you a good rationale analysis data in the near future. So single agent activity, ibrutinib can cause a response rate of 65 to 70%, but that's not enough. We want it to be higher. How about 100%? So we are in the way of, we need to constantly aspire for better efficacy. So during that, it is known that ibrutinib could drive the lymphoma cells from their tumor compartment into the peripheral blood compartment, what I call it the compartment shift phenomena. So we recapitulated, redemonstrated the compartment shift phenomena in mice model. So in mice model, after ibrutinib, the 5 out of 5 mice have dramatically increased lymphocytosis in the peripheral blood by Tillman blood analysis. And if we if we infuse the rituximab into the blood, where the rituximab will immediately work on the cells that were driven out of their home environment, and there's a dramatic improvement of beta 2, which dramatic decrease of the tumor volume as reflected by beta 2, and there's also a prolonged survival in mice. Based on the preclinical data and also the hypothesis of targeting the blood cells in the peripheral blood, we designed a clinical trial with ibrutinib plus rituximab. And I showed you yesterday the response, the side effect profile is very favorable, and 46 out of 50 patients responded to therapy, and for an overall response of 88%. But if you look, the 34 patients who has Ki-67 is less than 50, the response rate was 100%. The CR rate was reached all the way to 56%. So this data actually is guiding my practice at MD Anderson. I seldom use ibrutinib alone. I always use the combination. And if I also look at the Ki-67, if there are Ki-67 less than 50, I use the combination. The response rate continue to be. Now we have no exceptions to the rule. So we have improved the single agent 68 by adding another monoclonal antibody, reach the response rate unprecedented 88%. How about we put it in the upfront? So we designed the clinical trial where we would use only 50% of the chemotherapy hyper-CVAD if the patient achieved a complete remission with a chemo-free combination. In this case, is ibrutinib and ibrutinib. And we enrolled so far 36 out of 50 patients. We have a good data. I wanted to tell you preliminarily so far all the patients, reevaluated patients responded to the chemo-free part, and so far in a very short median survival. Follow-up of a few months, the CR rate already reached 60%. So I'm looking forward to share the data with you at ASH this year. So those are the single agent ibrutinib studies in relapsed mantle cell lymphoma. It has the 1104 study, it has the SPARK study, it has the European study where ibrutinib is compared with a tensor-limus, and then it has the John Bird combination study. This is the combination studies ever started in the mantle cell lymphoma with ibrutinib. You can see that we talked about the rituximab adding to ibrutinib. There's other studies to come as well. These are there for your reference. And so newer generation of the BTK inhibitors, ACP-196, as you can see, they claim in CLL is 95%. The phase 2 clinical trial with ACP-196 for relapsed mantle cell lymphoma has finished enrollment, and I think we will share some of the data at ASH with you. And the BGB study is a first one in human. It started in Australia. They want the U.S. side to be involved. They have this stage 1A design with different cohorts. And I have to point it out, the prior therapies is only 2 prior therapies. You can look at this. So if you look at the mantle cell lymphoma patients, they have about a few of the mantle cell lymphoma patients for a response rate of 80% so far. So it's not final. And MD Anderson has become a hub for mantle cell lymphoma research. This is I'm working on the moonshot, but this is my priority. So mantle cell lymphoma at this point In the front line, we finished the SHINE study, and we are looking forward for the interim analysis. We have an ongoing ibrutinib rituximab upfront study for frontline elderly patients. So if their Ki-67 is low, we use this one. But if the Ki-67 is high, we use the R2 ibrutinib. This one is also about to open very soon. And then we have the young fit patient with less than 65, and we have window 1, and we are working with the Janssen, with the Mark, and with other colleagues on the to have window 2, which will incorporate the venetoclax into the chemo-free regimen. So clinical trial for relapsed, we have antibodies like DART, which is a bispecific antibody, is a CD19 and a CD3, and we have daratumumab, and we work for Janssen partially. And so BTK inhibitors, we do ibrutinib, CP1X, BGB3, Southern 111, and checkpoint inhibitors. We have Nivo, we have Pembro, we have a growing alliances with Celgene, with other people to have checkpoint inhibitors, and for mantle cell lymphoma only. So targeted agents, so we have ONC201, first in human study, ER stress agent, PI3, venetoclax, carfilzomib, CAR T cells. We practically work with every CAR T cell company, including Kite, Juno, Sleeping Beauty, and Novartis, and then PI3 kinase is very important for idelalisib, divarasib, CUDC. And I'm also working on a personalized therapy with PDX tumors, and with the in vitro screening of thousands of agents, which then find the best agents and combinations and validate that in with the PDX in vivo data, and coupled with the molecular profiling. So I'm doing that to personalize, which is not on the list here. The trial is not open. So I wanted to see that MD Anderson is very committed for B-cell lymphoma. We have a strategy for mantle, for follicular, for large cell lymphoma, pretty much everything. And I just shared it with you, mantle cell lymphoma. Thank you. No questions.
On the trial you showed, you're doing ibrutinib, and after that you're doing hyper-CVAD?
Yeah.
Okay.
Yeah. And so usually hyper-CVAD is 6 to 8 cycles. So if we are able to, the trial is designed, the first 6 to 12 months is the only ibrutinib with rituximab. So if they achieve complete remission, we only use 4 cycles of hyper-CVAD. So this way we reduce the chemo by 50%. I really think we will get rid of the 6% of the late MDS and leukemia. That's
and if they don't achieve complete remission?
Well, they don't achieve complete remission in 6 cycles. Yeah.
So what's the in that your patients? What's the cure rate with our hyper-CVAD alone?
So the historic data we published the 10-year and 15-year follow-up. So for the 15-year follow-up, the overall survival, median overall survival is 10.4 months. 10.4 years. Sorry about that.
Right, but you're talking about cure. And if you act, and so
I never mentioned the cure with the hyper-CVAD. Okay.
Exactly so, right.
For the record, yeah.
So if you look at the long-term curves, they continue to fall.
So I guess I don't really
no plateau. Yeah.
I don't really understand your strategy then. You've shown, very effective treatment with ibrutinib or ibrutinib plus rituximab, and in fact you probably expect some CRs, right?
I expect the CR to reach 80%.
Yeah. Okay. Yeah. So why arbitrarily add on 4 cycles of hyper-CVAD or whatever? That seems very arbitrary to me, and it's not answering a question because it's not a randomized trial.
I totally think you're missing the point, Steve. So for the young fit patient, and the best therapy, the best result is either it has to something with the intensity. So for newly diagnosed mantle cell lymphoma, for a young fit patient, you either have to treat the patient with a Nordic therapy or the hyper-CVAD. There's Anna like he's from Mass General. They did this NCCN guideline. They find the R-top R-bendamustine is inadequate for young fit patients. You have to. They showed the overall survival is that it is identical to use either Nordic or hyper-CVAD. Okay, so but Nordic or hyper-CVAD, they are associated with the severe toxicities, acute toxicity, and especially the worst fatal late complications like MDS leukemia. So that's maybe one of the reasons why, the curve is not a plateau because they relapse and they also die from second long-term toxicities. So this is one of the reasons. I think it is too premature to treat the young fit patient, especially the aggressive disease, with a chemo-free regimen, because hyper-CVAD and Nordic therapy, chemotherapy are associated with a 10-year overall survival. You don't want to totally get rid of chemotherapy. It's not arbitrary. It's a progressive, stepwise way to reduce chemotherapy and late toxicity, so that eventually the final result is improved efficacy and decreased toxicity.
So, Michael, I actually agree that I do think that using these drugs alone, especially RI alone, and mantle cell up front is probably not the way to go. What I don't understand though is why don't you want to combine RI with hyper-CVAD up front? Because that way, to me, you can leverage the sensitization of the tumor by the BCR signal and hopefully get maximal cell kill and possibly cure. So I don't know why you're giving this long lead-in, quite honestly.
So I think there's many ways to skin the cat, and your way is the most conventional way. But I wanted to assure you that we want to do it this way because we call it the window study, a window of chemo-free therapy before chemotherapy, this chemo-free is not any less powerful than the hyper-CVAD. So far, response rate is 100%, and then the 60% of people after 4 to 6 cycles achieved complete remission, and those people had big tumors on the neck, big spleen, massive tumor load, and we're not. So it's a very powerful therapy.
Right, but when studies in combination with chemotherapy, like when they looked at when R follows chemotherapy, it's not as potent as given with chemo. So I'm not denying everything you said. I'm just saying that, and I'm a big fan of window studies. We usually do it for a very short period to get some idea what the drug actually does. But the question is, could resistance be coming up? But one thing I wasn't sure, when you do give hyper-CVAD, do you continue the ibrutinib?
No, we stop it. We stop it.
So that's the other thing is, why stop it?
So this is just the way that I designed it, the protocol, and it's not perfect. But then we are getting good results. So.
Excuse me. Any preliminary data on daratumumab?
Well, with this, so Mark, should I? Well, okay. So Dara is a fantastic drug for myeloma. I'm treating it even with a single agent. I frequently get very good results for very, very late advanced myeloma patients. Okay, so there's a clinical trial going on with the daratumumab by Janssen in non-Hodgkin lymphoma. Has 3 arms: follicular and large cell lymphoma, and actually CLL, and also mantle cell lymphoma. And so far, it's too early to tell the efficacy. Trials still going on. It's alone. It's yeah. All right. So when I'm in the future, when I design the window 2, 3 study, I'm going to come to NIH and You can give me all the.
We would be happy to have you.